Hello everyone, my name is Ilyes Baali. I'm a PhD student at the Computational Biology program at Weill Cornell Medicine School. First, I would like to thank the organizers for the opportunity to present this work. So today I'll be talking about a method that I have developed during my master studies called DriveWays to identify overlapping driver pathways in cancer. From cancer biology, we know that cancer is caused by a small set of somatic mutations known as driver mutations. Besides that, we have much higher number of mutations known as personal mutations. These mutations have no consequence for cancer. So for many years, researchers are trying to distinguish between these driver and personal mutations. So what they have done is they try to sequence the genome of many samples and compare the variations across tumor samples at the individual gene level to identify what's so-called significantly mutated genes. Researchers have come up with various ways to score the mutation frequency of the genes. So once we rank the genes based on their significance scores, we end up with a plot like this. So here in red we have significantly mutated genes, and in blue we have long tail of non-significant genes. So cancer cannot be explained by this small number of driver genes, and the long tail region is known to contain genes that are associated with cancer. So researchers started looking at interactions between the genes. Let's take A and B. A is significantly mutated and B is not significantly mutated, but A interacts with B to perform a functionality. So both of these genes are important for cancer. And what happened is that A is more frequently mutated than B. So researchers start looking at interactions between the genes and try to identify driver modules instead of driver genes. So there are a lot of methods that have been developed to identify driver pathways, but all of them make non-overlap assumption. This assumption is wrong, particularly in cancer, because many of the genes participate in different pathways. Now I'll start with providing mathematical information for this problem. We call this overlapping driver module identification in cancer problem. So given hemophilia network G and mutation data, let M be a subset of genes from G donating a module. We define a MS score for the module as the coverage of the modules times the mutual exclusivity. Both coverage and mutual exclusivity are derived from the knowledge of somatic mutation process. In cancer, pathways tend to be perturbed in large number of patients. Those driver modules should have high coverage, and it's defined as the fraction of samples that have at least the mutation in a gene from module M. And mutual exclusivity, it's observed that sets of genes that are commonly involved in the same cancer pathway tend not to appear mutated together in the same patient. And the genes in module M should be mutually exclusive across the samples. So our goal is to find the set D of possibly overlapping modules that maximize the sum of the MS score of the modules, and this score is called ODMSS. And the set D should satisfy the following conditions. First, the connectivity. Each module G is connected, and all the modules in D are unique, and each of the modules has a minimum size delta M, and the total size, so the sum of all the genes in all the modules is delta M. We showed that the computation complexity of this problem is NP-hard, and it has pseudo-polynomial time solution from constant delta M. For more detail, please refer to the paper. Now I'll provide a heuristic algorithm for solving such a problem. We call this algorithm DriveWays. So this algorithm follows a two-step 3D seed and extend procedure. The first step is to rank the seed, and the second step is to construct a set of driver modules. So the first step is to find the order at which the genes are seeded. So we filter out all the genes that are mutated in less than 1% of the cohort, and then we sort the genes based on this score, which is MS of the neighbors of G. So MS is coverage of the module times the mutual exclusivity, and N of G donates the set of neighbors of G. So this score measures how fit the gene is for further growth. And the second step is to construct a set of driver modules. So the way we do that, so first we start with ranked seeds. So here we have our set D. So the first module is grown out of the red seed, and the second one from this brown one. And so because the green is part of the second module, we skip growing the third seed, the third module from this seed, and we go to the blue. So here we have our current module, which is module 3. Now I will explain in detail how we do the growth process. So first we look at the neighbor of this module, so all the genes that are incident in a gene in the module, and we filter this neighbor to come up with a set of candidate genes. These candidate genes should satisfy these two conditions. The first condition is related to the coverage of the module. So the coverage of M with any gene should be more than T times coverage of the current module over the mutual exclusivity. So this enforces lower bound for the update coverage value of the module when GI is inserted, and T controls the size of the module in indirect way. So when we increase T, we make it hard to include more genes. The size of the modules is going to be small, and vice versa. And the second condition is related to the overlap between the modules. So for each gene GI, the degree of GI with respect to M, that's the numbers of GI in module M, over the mean degree of GI across the modules in D that GI is a member of, should be greater than 1 over D. So D serves as a mean to control the overlap rate between the modules. So as D decreases, genes that are part of previous modules are not going to be considered for the next steps. And in that way, the overlap between the modules will be small. So both T and D are set using a Bayesian optimization procedure. So now we have our current module and the candidate set of genes. So the first step is consider adding a gene. So we construct this new module MA by adding a gene such that the MS score is maximized. And the second module is MR. We consider removing a gene from the current module such that the MS score is greater. So then what we do is we compare the MS scores of these two with the current module, and which of the two has higher score, that modification is committed. And if none of them has higher score than the current module, then the process is stopped, and we look at the size of the current module. If it satisfies the minimum size requirement, then it's going to be added to the solution set D, and the whole process is stopped once we reach the requirement of total size data S. So now I'll talk about the results and some discussion. So the method we compare against are 4 methods. So 2 of them provide non-overlapping modules, which are hotnet to MX world, and 2 methods that provides overlap modules. So cluster 1 is not designed for this specific problem, but it's a general community detection in graphs. And MumCover provides overlapping modules as post-processing step. The input to all the methods consists of PPI network construct from intact network that contains 8,000 genes in 83 edges. Some of the edges were filtered out if they have confidence value less than 0.35. For the mutation data, we use TCGA pan-cancer cohort that contains 11,000 genes and 3,000 samples. And as for parameters, we use default parameters for all the methods. And for DriveWays, we use .m, that's small, the minimum size as 3, and .s is set according to the reference pathway's total size. So for the first evaluation, we look at output as union of all the genes, and we call that evaluations omitting modularity. So what we do is we run the algorithms to output 100 to 1,000 unique genes in incremental step of 100, and for each output, we look at the overlap with CGC genes, and then we plot this for curve. So as you can see, DriveWays has a higher area under the curve, so it was doing a good job at recovering known constant drivers. And the second set of evaluation looks at the output at the module level, and this is more important than looking at the output as the union of all the genes. So we call that modularity specific metric. So the first metric is our objective function, the MS score, which is the sum of MS scores of the modules in D. So in this plot, in the y-axis we have the MS score, and in the x-axis we have experiments for 3 different total size data S. And the way we calculate this data S is using 3 reference pathways, which are KEGG, Reactome, and BioCarta. for each reference pathway. We filtered the pathways to include only cancer-known genes. So we use a CGC set, and then we kept any resulting pathways with size greater than delta M, and then we calculate the total size of the resulting pathways. So as you can see here, DriveWays outperformed all other methods with respect to this metric. And the second metric is maximum matching ratio. So this evaluates the functional coherence of the predicted modules. The way we calculate that is we construct this bipartite graph containing predicted modules on one side and the reference pathways on the other side. So we give weights to the edges between predicted and reference modules using three similarity measures, which are overlap ratio, hypergeometric test Q-value, and Go consistency score. And then we find the maximum matching graph, which is shown here with the thick edges, and then we take the average edge weight of the maximum matching graph as the score for this. So here, each of the plot is for a similarity measure. In the y-axis, we have the MMR score, and in the x-axis, we have the reference pathways. So as you can see here, DriveWays outperform all the other methods except for Go consistency similarity measure for Reactome, where it has tied with memcover method. We define the precision score as the average of best match scores for each of the predict modules, and recall as the average of best match scores for the reference pathways. So using the precision and recall scores, we calculate F1 scores. So here's a summary of the scores for using different reference pathways and similarity measures. So DriveWays outperform all the methods in respect of F1 score. So some biological insights. So looking at the top 10 modules in context of MMR, 5 of these modules directly correspond to cancer pathways, and 2 of them are associated with pathways related to cancer. Another interesting match is observed between our 7th ranking module and bladder cancer pathway. So our predicted module contains 6 genes, 4 of which appear in the bladder cancer pathway. VHL, a well-known chemosuppressor, is among the 2 genes that appear in our predicted module but not in bladder cancer pathway. Interestingly, among the cancer types in pan-cancer cohort, bladder cancer ranks second after renal cell carcinoma in terms of VHL's mutation frequency. So looking at the top 20 predicted modules in context of precision, among the best matched pathways, we observed pathways in cancer and other signal pathways that are associated with cancer. We were able also to find potentially novel cancer drivers, which are LTBP1 that appear in TGFB signal pathway, SMC3 and SMC1 that appear in cell cycle, and FLN8 that appears in MAPK signal pathway. Also looking at the cancer genes that are in multiple pathways, we found an RG1 that was only identified by our method, and none of the method was able to find this gene. So all the previous results were shown where we used TGCA pan-cancer cohort and CGC genes as reference genes. So we repeat the same procedure where we only select BRCA samples and we used CancerMine BRCA genes as a reference set. So looking at top 20 predicted modules in context of precision, we found a number of potentially novel cancer drivers, which are PIK3R1, CDK4, CDK6, and SF3B1. So interestingly, PIK3R1 was found in a model containing both PIK3CA and ERBB2, both of which are associated with breast cancer from CancerMine reference set. It's noteworthy that the mutation frequency of PIK3R1 in the cohort is quite low. Only 19 of the samples contain mutation in this gene. So this demonstrates the effectiveness of the proposed model of DriveWays in detecting rare drivers through the insightful incorporation of mutual exclusivity concept. So in conclusion, we provide the definition of an optimization problem for overlapping modules. We provide an algorithm for that called DriveWays. that controls the amount of overlap between the modules in a flexible way. We also introduced a number of evaluation metrics that are suitable for this problem. And we find that the identified modules are enriched for known cancer genes and also enriched for correct pathways. So for acknowledgement, I would like to thank Gisem Mertan and Hilal Kazan for their help during this work, and also all the members of Antalya Computation Biology Lab. And I would like also to thank the sponsor for their sponsoring. Thank you. And if you have any questions, thanks.